A normal acute wound follows a predictable, high-speed schedule. Within minutes, bleeding stops. Over the next few days, inflammation clears debris, and by week 3, the tissue is largely restored. In chronic wounds, this timeline simply stops, leaving the injury open and vulnerable for months. Stalled healing is frequently attributed to external factors, like poor hygiene or persistent bacterial infections. We tend to blame the environment for the body's failure to close the gap. The source of this failure is often located deep within the genetic code. Microscopic errors in our DNA can prevent the regenerative process from ever reaching the surface of the skin. To understand how a genetic typo can stop a wound from closing, we have to look at the specific biochemical sequence required for a successful repair. The physical ability of our skin to heal is dictated by invisible sequences occurring at the molecular level. Standard tissue repair relies on 5 sequential phases: hemostasis, inflammation, proliferation, contraction, and remodeling. Hemostasis begins the exact moment of injury. Blood flow carries a massive influx of platelets directly to the damaged tissue. While platelets aggregate to form physical clots to halt blood loss, they primarily function as dynamic chemical signaling hubs. These cells establish the foundational scaffolding for the wound, releasing the specific instructions that dictate the timing of the immune response and subsequent tissue growth. Serotonin production is almost entirely peripheral. It induces short-term swelling in the vascular endothelial cells, creating physical gaps in the vessel walls. These newly formed gaps allow critical defense cells, such as neutrophils, to migrate out of the circulation and into the damaged tissue. This serotonergic signaling acts as the primary molecular trigger for inflammation, allowing the healing timeline to proceed to the next stage. The source of this signal is found on chromosome 13, the location of the 5-HTR2A gene, which provides instructions for building the platelet serotonin receptors. In the 102TC polymorphism, a single error occurs in the sequence. A thymine nucleotide is replaced by a cytosine at position 102. This mutation is considered silent because it doesn't change the protein's physical structure. Both versions encode the amino acid serine, so the resulting receptor shape remains perfectly normal. However, the genetic shift causes a significant drop in gene expression. The total number of receptors displayed on the platelet membrane begins to thin out. With too few receptors to bind with, a high concentration of unbound serotonin is left sitting in the local plasma. This excess triggers a rapid cleanup response. SERT transporters pull the serotonin away, and MAOB enzymes dismantle the molecules, removing them from the site. This accelerated breakdown creates a localized paradox. The wound site, which needs serotonin to trigger inflammation, is left entirely depleted of the chemical. Without that serotonergic signal, the inflammatory response fails. Immune cells cannot enter the tissue, and the 5-stage healing timeline stalls indefinitely. A single nucleotide shift on chromosome 13 can effectively stall the body's entire regenerative sequence. Mapping this genetic bottleneck allows for the development of personalized therapies. By identifying where the signaling fails, we can find ways to bypass the deficiency and restore the body's ability to close chronic wounds. If you enjoyed this video, please give a like and subscribe. And what would be really helpful would be some suggestions for future videos. Today's video was brought to you by Synestec Systems, LLC, founded by Shane Persinger.